Good evening, everyone. My name is Dawn Laprette, and I am one of the program managers at the Chicago Council on Science and Technology. Thank you for joining us for today's program, Ask the Scientist 2020 Nobel Science Topics. This program is presented to you by C2ST in partnership with the Carl R. Rose Institute for Genomic Biology and the Kleinmet Center for Genomics in Business and Society from the University of Illinois, Urbana-Champaign. If you are new to C2ST, we are a not-for-profit organization founded in 2007, working to enhance the public's understanding and appreciation of science and technology and its impact on our lives. Please visit c2st.org to learn how you can donate and get information about upcoming virtual programs. You can also sign up for our mailing list so that you can get reminders about these programs as well. So on to some housekeeping. Please type c2st2.cnf.io into your browser and click today's session to ask questions and to evaluate the program at the end. We would sincerely appreciate your feedback if you can complete the survey after today's program. You'll be able to ask questions while you're watching. Our staff will be checking C2ST TV on YouTube and Facebook Live for your questions. If you want to ask questions directly, please go to c2st2.cnf.io. The format for today's program will be to briefly introduce the panelists, allowing each approximately 10 minutes to offer a bit of background on their area of expertise. We will begin the Q&A at around 5:30, 5:35, and wrap up today's program by 6 p.m. I'd like to briefly introduce our stellar panelists for today in order of presentation. On the topic of black holes, we have Nicolás Yunes, who is a professor of physics at UIUC and the founding director of the Illinois Center for Advanced Studies of the Universe. He is a leader in gravitational wave observations to test Einstein's theory of general relativity systematically, and he is an internationally recognized expert in other aspects of gravity, including black holes, which you'll hear about today, and neutron stars. On the topic of CRISPR, we have Thom Gaj. He is an assistant professor of bioengineering at UIUC. His research lies at the intersection of neuroscience and engineering. He focuses on developing new technologies to facilitate the correction and treatment of neurodegenerative diseases such as ALS and Huntington's disease. And last but not least, on the topic of hepatitis C, Claudia Hawkins is an associate professor of medicine and infectious diseases, global health faculty member, and the director of the Division of Infectious Diseases, Viral Hepatitis HIV Co-Infection Program at Northwestern University. Her research focuses on the epidemiology of HIV and viral hepatitis B and C co-infection. She has collaborated globally with investigators from the US, Tanzania, Nigeria, Brazil, Thailand, and South Africa. Doctors, we are thrilled to have you join us today to shed light on the topics that were awarded the prizes in October. And Dr. Yunes, we would like to begin with you.
Thank you very much. So let me try to share screen, see if technology will cooperate. Then go to a little presentation that I have put together for you all to try to explain a little bit what was done in order to earn the Nobel Prizes this year, as well as in 2017. It all surrounds the idea and the concept of a black hole. So let's begin then with that, with the concept of a black hole. So what is a black hole? So one of the cornerstones of Einstein's theory of general relativity is that space and time form a type of continuum. And I'm representing that here through these great lines. And what's really interesting about Einstein's theory is that space and time can curve. They can stretch when in presence of dense objects like planets or like stars. Because you see, gravity to Einstein was not a force like it was for Newton that would just attract to different objects with mass. Rather, to Einstein, gravity was just a manifestation of the curvature of this space-time. Gravity, or the force of gravity, is just a consequence of bodies trying to move in as straight a path as possible, but not on a flat space and time continuum, but rather on a curved continuum. So the idea of an absolute time disappears. This was all very new, of course, in 1915 when he published, 1915, 1916, when he published these ideas. But today we have tremendous number of experiments that have verified all of these predictions to great detail. And perhaps one of the most astonishing prediction is that of a black hole. So as I told you before, when you take matter and you place it in space-time curves. But if you put enough matter in a small enough amount of space, space-time compresses so much or stretches so much that it forms a black hole. So if I take all of the mass in the sun and I compress it into a ball with a radius of about 30 city blocks in Chicago, I actually looked up how long a city block in Chicago is, then if you were able to compress all that gas that's in the sun into such a small sphere, then you would form a black hole. So a black hole is an object that is just incredibly dense, so dense that it produces an enormous gravitational field. And the field is so strong that nothing can really escape its interior. If you were to try to shine a flashlight from the edge of a black hole, light would actually not escape. It wouldn't travel straight out of your flashlight. It would actually fall back into the center of a black hole. That's why we call black holes black. So this is a consequence of general relativity that's generic. And the big question was, do these objects really exist in nature? And what happens when objects like this collide with each other? Do they collide in nature, in the universe? And this is what the Nobel Prize was given for in 2017. It turned out that 4 billion years ago, roughly, two black holes with masses of about 30 times the mass of our sun in each black hole slowly spiral into each other, eventually reaching velocities of about half the speed of light, merging and producing vibrations in space and time. These vibrations travel at the speed of light, and 4 billion years later, they reached Earth. And they reached Earth roughly at the same time as humans had built the first detectors of gravitational waves. And they had turned them on. These detectors, the LIGO and Virgo detectors, are laser interferometers. They're Michelson-type interferometers. And they can detect really tiny vibrations or stretching and compressing of distances. Of course, the effect is not as exaggerated as I just showed in that animation, but rather the changes in travel in the light travel time, in the amount of time it takes light to travel from one mirror to the next, changes by a tiny amount. 1 part in 1,000 the size of a nucleus. And this was measured in Washington State and in Louisiana in 2015. And this interference pattern that was observed could then be tracked, analyzed, and interpreted as a gravitational wave that had gone through Earth as produced by this cataclysmic merger of black holes. So that gravitational wave detection, this was the first time that we detected gravitational waves. Einstein had predicted that in the around 1920s or so. And not only was this the first detection of this, the last prediction of Einstein's theory that had not been verified yet, it also gave us direct confirmation of what happens when two black holes collide. Since then, the LIGO-Virgo collaboration has detected over 50 events of merging black holes and merging neutron stars, giving birth to the field of gravitational wave astrophysics. More recently, and here is a small animation. I guess I don't know if you're going to be able to hear the sound, because I think I didn't click on sound properly, but you can see the evolution of the frequency and time and the actual data in time. So this is actual data. What you're seeing here, it's not just an animation like or a simulation like I showed you earlier. But now, in 2020, small typo here, 2020, there was another set of observations. Actually, these observations had been going on for a very long time. It's just very hard to see what's really going on at the center of our own Milky Way. So what astronomers were able to do is develop these new optics and these new telescopes and really resolve stars, the motion of stars moving very close to the center of our galaxy. And we were able to track these orbits of these stars. And by being able to track these orbits, they were able to calculate the mass of the object that had to be at the center of our galaxy in order to make all of these stars go around it. That object at the center of our galaxy has 4 million times the mass of our sun. Yet when you shine telescopes to this region, you don't see the light of a star that's 4 million solar masses. You see nothing. So the only object in nature that we know that can do that is a black hole. It's the only object that can be so massive yet so compact to not produce light. So this is what the second Nobel Prize was given for the observation of supermassive black hole at the center of our galaxy, and also the verification that black holes are a natural consequence of Einstein's theory that is in some sense unavoidable. So with that, I will stop. Thank you, and I'll take any questions you have at the end of this. You're muted.
Thank you very much. That was a really great, concise way of talking about it, and I really appreciated the visuals. Dr. Guy, we're going to move on to you and talk about CRISPR.
All right. So screen should be shared. So thank you so much for the invitation, and I'm really delighted today to have a chance to talk to you all about CRISPR-Cas9 technologies, genome editing at large, and its application for enabling human gene therapy. So CRISPR-Cas9 is a technology that's most commonly used for gene editing that was awarded the Nobel Prize this year in chemistry. So today I want to talk a little bit about what gene editing is, how CRISPR-Cas9 has really sort of transformed or revolutionized the field of gene editing at large, and how we're using it to treat disorders in the future, hopefully. So any discussion that centers around CRISPR-Cas9 technology first is really going to have to start with what exactly is genome editing, sort of as we know it. And when we're talking about genome editing, we're really talking about the idea or concept of introducing targeted breaks in DNA. And the reason that this is the basis of gene editing is because gene editing is actually the repair outcomes introduced at that break by the cell. So if you introduce a DNA break within a DNA sequence, one of two things can happen. If you just introduce the break by itself, you get an activation of a DNA repair pathway within that cell that introduces really random base sequences at the point of that break that have the potential to turn off the expression or production of the proteins encoded within that gene. If you introduce a DNA break alongside another piece of DNA that matches your target sequence, and this is in biological terms known as homology, you then get a repair outcome known as homology-directed repair. And what happens there is that second piece of DNA actually gets copied or inserted into the point of DNA where you introduce that DNA break. And this is a repair pathway that's really powerful because it offers you an opportunity to correct mutant genes. And so if you have gene mutations that lead to the production of toxic mutant proteins, you can introduce a DNA break within that gene to turn off its production to hopefully eliminate the presence of that mutant toxic protein. And if you have gene sequences that carry mutations in them and you want to correct them, then you just introduce a DNA break and co-introduce, along with the tool to target that break, a homologous donor template that can then mediate correction. But the really central question to all of this is, how do you introduce or target these DNA breaks within very specific sequences? DNA breaks happen within cells randomly through UV damage, and they're generally bad. But if you can target them at very specific sites with programmable technology, then you really have the capacity to replace, correct, or delete very specific sequences of DNA. And what CRISPR-Cas9 is the technology that really enables that. So it really is a revolutionary genetic scissor-based tool. And so Jennifer Doudna and Emmanuelle Charpentier won the Nobel Prize this year for their work in describing the mechanisms of DNA targeting by an enzyme known as Cas9. And this is part of the name CRISPR-Cas9 that I think so many people have heard of. So what was really profound within their study that described the mechanism of DNA targeting is that it suggested an approach to use this system or reprogram this system to recognize virtually any DNA sequence that you want or DNA site that you want within large genomes with very minimal work or very minimal engineering. And so what exactly is CRISPR-Cas9 and why were they Jennifer Doudna and Emmanuelle Charpentier and their group studying this technology? Well, we've known about CRISPR-Cas9 systems for a while because we came to believe that they actually had evolved to provide bacteria and other microorganisms the ability to fight viral infections. So in a sense, CRISPR-Cas9 is an immune system within very simple microorganisms. And how it functions within these cells is really elegant. And so this is going to kind of be a three-step schematic that sort of illustrates that. Here we have our prokaryotic cell that's actually being invaded by a virus. This virus injects its DNA into the host cell in hopes of hijacking the host cell's machinery to produce more of the virus. But a prokaryotic cell with a CRISPR immune system has the ability to sense this foreign DNA and then insert it within its CRISPR genomic locus, so that when the prokaryotic cell is infected again and a second time by an invading foreign DNA, it will remember these foreign DNA sequences that it be integrated into the CRISPR locus, produce RNA sequences that then are complementary to the DNA encoded within these viruses and use their own enzymes, in most cases enzymes known as Cas9, to target and degrade this foreign invading DNA, thereby fighting off future infections through an immunological memory response. So both the groups of Emmanuelle Charpentier and Jennifer Doudna really elegantly repurposed this system for genome editing. And so how exactly does CRISPR-Cas9 technology work for gene editing? Well, in its simplest form, it consists of just two components. There's the Cas9 enzyme, kind of shown here as this yellow blob, and a guide RNA molecule that guides the Cas9 to specific DNA sequences. This guide RNA molecule is what's re-engineered every time you want to target a new DNA sequence. So if you're interested in a specific 20 base pair or 20 nucleotide site, for example, within gene X, you take that 20 base pair sequence and you actually insert it within this guide RNA, so that when you produce both of these molecules within a cell, they complex together and the Cas9 is directed to that specific genomic site because it's complementary to the guide RNA, and the RNA is going to bind to the DNA, as RNA and DNA tend to do. Upon RNA DNA heteroduplexation, or hybridization as it's known, the Cas9 will then become activated and then introduce a DNA break. And it's the presence of this DNA break, like I said earlier, then triggers the gene editing pathways within the cell. And if you introduce the Cas9 enzyme by itself with no guide RNA, then you can potentially disrupt the expression of a mutant gene. And if you co-deliver another piece of DNA that actually matches this target sequence, then you can do gene corrections. And the really powerful thing about CRISPR-Cas9 is that it really democratized gene editing. So before the use of CRISPR-Cas9 for gene editing, it was very challenging and technically laborious to actually create new gene editing proteins, because we had to engineer the proteins every time entirely from scratch to recognize new genomic sequences. And it can take anywhere from 6 months to 2 years to create new gene editing proteins. Using CRISPR-Cas9, you can now redirect it to new sites just within a matter of days. And so it's really efficient and really versatile and really profound in its ability to bring gene editing to the masses. And so because of these properties and because of its potential to correct mutations, CRISPR-Cas9 technologies hold really tremendous potential for human gene therapy. And there's really two types of way of using the technology for gene therapy. You can do something that's known as ex vivo gene editing, where you can take cells from a patient with mutations, for example, and then take those cells, treat them with your gene editing systems to correct mutations, expand them, and then potentially transplant them back into the patient to replenish that population of cells that you targeted. Alternatively, you could actually also do gene editing in vivo within a whole tissue of an organism. This is a little bit more complicated and probably will take a number of more years to actually see the clinic, but there's a number of really promising clinical trials actually ongoing right now for this concept of ex vivo gene therapy, for example, for sickle cell anemia. What our laboratory is actually interested in is using these gene editing technologies to treat progressive neurodegenerative disorders. And one such type of progressive neurodegenerative disorder that we're really focused on developing new therapies for is ALS, or Lou Gehrig's disease. And ALS is unfortunately a currently incurable disorder with very few treatment options that involves the progressive loss of motor neurons both within the brain and the spinal cord. And it's the loss of motor neurons over time that eventually will lead to paralytic features, respiratory problems, and unfortunately death within about 3 to 5 years of symptoms onset. And so ALS is a very heterogeneous disorder. There are both inherited forms and spontaneous forms of the disease. We're most focused on developing corrective gene therapies for inherited forms of ALS, and particularly the most common forms, those known to be caused by mutations within a gene known as C9orf72, or within mutations of a gene known as SOD1 or SOD1. And specifically in the case of SOD1 ALS that we've had a lot of early success developing new gene editing therapies for, we've developed strategies to target the mutant SOD1 gene to disrupt its expression. And so here's just a kind of a little bit of a sneak peek of the results of our ongoing work. So we've developed CRISPR technologies to specifically recognize mutant SOD1 genes, so that when cells that are expressing the mutant protein that causes ALS are treated with the CRISPR nucleases, we actually decrease the amount of mutant protein present within the cell. And we do a number of high-throughput genomic studies to actually quantify and confirm that the loss of mutant protein is actually permanent and the product of gene editing. And we get this in frequencies of about 30 to 50% of all cells that are introduced with the gene editing protein. Our ultimate goal is to convert this into a gene therapy that can really benefit ALS patients. And in order to actually bring it ultimately to the clinic, that means testing out our approaches within animal models that have been engineered to develop features that mimic the disease in humans. And so we've administered these technologies into ALS mouse models, and we found that they're able to be produced at really high efficiencies within the cell types that drive the disease. And in this particular form of ALS, it's motor neurons that die, but it's actually surrounding glial cells or surrounding cells known as glial cells that drive the death of these motor neurons. And we found that we're able to deliver our CRISPR cargo to these cells at really high efficiencies, between 70%, and now we've actually improved on this, and we can actually target upwards of 90% of these mutant cells within the spinal cords of these animals. And we found that when the animals are actually dosed with these CRISPR therapies, they demonstrate a number of very significant therapeutic outcomes. We found that these animals have improved survival, a dramatically slowed disease progression, which is really important. We administer these therapies at the point that these animals are starting to first show the signs of disease. And once administered, they actually then develop the disease much more slowly. And we measure this in terms of their motor function over time, and also their muscle atrophy. So we quantify this, and we find that animals that are treated with gene editing have dramatically reduced muscle atrophy over time. And then when we analyze animals after they eventually succumb to the disease, because this is ultimately fatal, and the mice models that we use are very aggressive, we found that the animals treated by gene editing have dramatically reduced mutant proteins all throughout their entire spinal cord. Which is very exciting, suggesting that the tools that we're delivering are able to reduce the protein that we all suspect is toxic and driving pathology in this particular case. And so CRISPR-Cas9 technology has really revolutionized the fields of gene editing, largely because of its flexibility, how efficient it is, and how versatile it is. It can do a lot of things beyond just simply introducing DNA breaks. And so it's become really a scaffold for a number of high-throughput genomic applications. Our research team and others are really actively engaged in developing new therapies that harness the potential of CRISPR-Cas9, both traditional Cas9 gene editing technologies, and also now a number of next generation tools that are able to carry out more sophisticated forms of gene editing. So there's a lot of promise and potential with CRISPR-Cas9 and other gene editing tools, but there are a number of key questions that the field is going to have to answer within the next few years. What are the risks for expressing or producing the Cas9 protein within cells? Does its long-term presence within cells lead to mutations at unintended sites? And are those potentially going to be counterproductive to our therapeutic intent? And what are the types of immune responses to the Cas9 protein? This is a protein that's bacterial in nature, and so there's a lot of concern within the field that producing this protein long-term can actually elicit adverse effects within cells because it may be recognized as foreign. And so this is a technology with a lot of exciting potential. Our group and many others are hoping to advance it to the clinic within the next 5 to 10 years. But there's a number of very serious scientific questions that we're also hoping to answer along the way. So really quickly, I presented some results, and a huge thank you to Colin Lim, a fantastic graduate student within our group, and a former postdoc named Alex Brooks, who helped push forward our initial work on ALS and CRISPR-based therapies for it. So thank you so much, and I'm looking forward to answering any questions that may come up. I think you are muted.
Sorry. Thank you very much. I really appreciate you presenting that topic in a way that we can understand. And wow, I can't imagine the benefit of taking something that was 6 months to 2 years and doing it in days. Fantastic. Our next topic is hepatitis C, and I'd like to invite Dr. Hawkins to share some background on that topic.
Okay, so thank you to C2ST for inviting me, and I'll just share my screen here. Can everyone see those?
Yes.
Yes, great. Okay. Alrighty. So, well, thanks again to CCST for inviting me to talk about hepatitis C and our Nobel laureates. So in October this year, as you all know, the Nobel Assembly awarded the 2020 Nobel Prize in Physiology or Medicine jointly to Harvey J. Alter, Michael Houghton, and Charles Rice, who are 3 scientists who've made a decisive contribution to the fight against hepatitis C, which is a major global health problem that causes cirrhosis and liver cancer in people around the world. So their combined efforts in the discovery of hepatitis C virus made it possible for blood tests and new medicines to be developed that have saved countless millions of lives and set us really on a now very realistic path to complete elimination of this deadly disease. So what is hepatitis C? So hepatitis C is a type of viral hepatitis caused by the hepatitis C virus that can cause either acute or chronic infection, primarily affecting the liver. So the most common modes of infection are through exposure to small quantities of blood, and examples of the common modes of transmission are seen on the right of this slide and include injection drug use, which are the most commonest modes of transmission in Europe and the USA, and then unsafe injection practices, and then unsafe healthcare practices and transfusion of unscreened blood and blood products, which are typical or common in low and middle income countries. So about 80% of people who acquire hepatitis C will actually go on to develop chronic infection. So i.e. their body's own immune system doesn't clear it. And hepatitis C then causes inflammation in the liver, which eventually progresses over 10 to 30 years if left untreated to fibrosis and cirrhosis and end-stage liver disease, and in some cases, liver cancer. So the bad thing about hepatitis C is that few people experience symptoms in the first few decades after infection. So it's why it's known as the silent killer, as people don't really become symptomatic until the late stage or complications occur. And for these reasons, it's estimated that about half of the population with hepatitis C actually don't know they have it. So how widespread is it? So hepatitis C is a global problem. So globally, an estimated 71 million people are living with chronic hepatitis C infection. There are about 1.75 million deaths per year, new infections per year. And about 500,000 people die from chronic hepatitis C liver complications each year, which in contrast to malaria, TB, and HIV, other well-known infectious disease killers, have been rising in recent years. And some of the most heavily affected regions in the world, as you can see on this map, in the darker areas, are Eastern Mediterranean region, Europe, and which run prevalence rates around 2.5 to around 2.3%. In Egypt, one of the worst affected areas, about a quarter of the population is infected. And in sub-Saharan Africa, where I work, as well as other low and middle income countries have limited diagnostics for hepatitis C, the true prevalence is really not very well known. And some of the work I've been doing in sub-Saharan Africa has been characterizing hepatitis C in certain populations, as well as examining the burden of liver disease in patients with viral hepatitis in these regions. In a recent study in Nigeria we conducted, we were one of the first groups to conduct viral load testing for hepatitis C in adults with liver cancer, which is a big problem in Nigeria, and found about 2/3 of them had active hepatitis C infection, and most of those cases were undiagnosed. So what about closer to home? So in the US, it's estimated about 2.4 million people are living with hepatitis C. Some of the highest rates of infection are in the birth cohort, so people born between 1945 to 1965, although we're seeing increasing numbers of new infections in younger age groups, and primarily due to increased use of injection drugs, and rising numbers among our minority groups as well, as shown in this graph on the left. Around 15,000 people with chronic hepatitis C complications will die each year in the US. And again, worth noting that there's a big minority distribution, with certain minority groups having much higher death rates than others. So is there a cure for hepatitis C? And yes, there is. And so this is one of the big topics, bigger revolutionary areas. So in 2013, new oral medications called direct-acting antivirals, these shown on the left, that target 3 of the main proteins involved in hepatitis C viral replication, were developed that could rapidly cure over 95% of people with hepatitis C with just 8 to 12 weeks of oral therapy. So this was considered, as I said, a revolution in the treatment of hepatitis C, because until this time, all we had available were therapies that were injection form only, very toxic, and offered a very minimal chance of cure. And so the development of these DAAs was really considered a watershed moment in hepatitis C. And I can say, as an infectious disease specialist, curing hepatitis C has been one of the most rewarding aspects of my career to date. So how did we get here, and what roles did our Nobel laureates play in these significant advances? So it all started back in the 70s when Harvey J. Alter was at the NIH and was studying the occurrence of hepatitis in patients who had received blood transfusions that couldn't be attributed to hepatitis A or B, tests for which were both available by that time. And he and his colleagues were able to prove that blood-to-blood transmission of an undiscovered agent, and that this agent likely had the characteristics of a virus, and they subsequently named it as non-A, non-B hepatitis. About a decade later, Michael Halton worked for the pharmaceutical firm Chiron and was able to successfully identify cloned viral DNA fragments encoding viral protein, and showed that this clone was derived from a novel RNA virus belonging to the flavivirus family, and then it was named hepatitis C. But that wasn't the end. The discovery was obviously decisive of the virus, but one essential piece of the puzzle was missing, meaning could that virus alone be causing the hepatitis? And this is a question that Charles Rice answered, a researcher at that time at Washington University in St. Louis, who went on to clone a variant of the hepatitis C virus, which he injected into chimpanzees, observed changes in the liver and the blood consistent with chronic infection, and thus proving that the virus alone caused liver complications. So all this work was incredibly significant, leading to sensitive blood tests to diagnose hep C, prevention interventions such as screening blood donations, which led to significant reductions in cases, and obviously the rapid development of curative antiviral drugs. And as said by Stephanie Langle, a virologist at Duke University, that's really a story of investing in basic science and having it pay off later on down the road. So the big final question remaining is whether with all these advances we can really truly ever eliminate hepatitis C, and my hope is we can. So back in 2016, recognizing that a major public health activities are needed to combat viral hepatitis worldwide, the World Health Assembly adopted the Global Health Sector Strategy to eliminate viral hepatitis by 2030. They set some very bold targets, shown on the left here in the graph, to decrease the number of new cases of hepatitis C by approximately 90%, and reduce the number of hepatitis C-related deaths by approximately 65%. And they were trying to achieve this through a combination of preventing transmission by improving blood safety and infection control measures, particularly in high prevalent areas in low and middle income countries, extending harm reduction services aimed at reducing transmission among people who inject drug use and who inject drugs, and expanding and testing and the direct acting antiviral treatment for those who are already infected to reduce overall mortality. So much progress has been made by several WHO member states. However, there are significant elimination challenges with many of these strategies that people like myself and others working in the field continue to work on, including improving hepatitis C diagnostics with new technologies, improving screening and linkage to care, especially among the harder to reach populations, and raising awareness. And finally, on a global level, the lack of political prioritization, insufficient funding for Hep C treatments, and lack of access are all significant challenges that will need to be addressed by all countries if complete elimination is ever to be achieved. So with that, I'll stop and look forward to taking questions.
I am muted this time. Thank you, Dr. Hawkins. Again, another disease where we see underrepresented groups and poor groups being affected unfairly. Thank you all for your information. We do have a number of questions. It's about 5:38, so we have about 20 minutes left for questions. And so, Dr. Yunes, I'm going to start with you. And one of the questions that came up was, are there any potential dangers or consequences of the black holes merging?
Yeah, that's a great question. No, there are no dangers to people here on Earth. The effect of gravitational waves is very, very tiny. So gravitational waves, as they go through us, they're constantly being produced in the universe. So they're constantly going through our bodies, they're going through Earth. They change the separation between our atoms by a tiny, tiny amount. So we don't have to worry about it. The only way that we would be able to really hear a gravitational wave and it would affect visibly our bodies is if two black holes were to collide and merge in our solar system, like roughly where the moon is, or between where the moon is and where the sun is. But that's just impossible. Space is huge and black holes are really tiny, and there are not that many of them. Well, astrophysically speaking, I think we're safe.
Thank you. Thank you very much. Dr. Gaj, I'm going to ask you a question regarding how Cas9 is introduced or injected into the cell.
Yeah, that's a good question. And that's sort of the fundamental barrier behind its ultimate success for gene or cell therapies. How you deliver the Cas9 enzyme first is going to be dependent on what your therapeutic approach is. So if you are interested in these types of ex vivo strategies where you're first going to take cells from a patient, then you have actually a wider range of delivery options available to you that are oftentimes higher efficiency. And so efficiency means the number of cells that receive the Cas9 protein. So for these ex vivo strategies, you can use techniques that are known as electroporation, where you introduce electric currents into the cells to kind of create pores or membranes where you can introduce then DNA or the Cas9 protein itself into them. The DNA then expresses the Cas9 gene, and you can have editing within those cells. It's a little bit more challenging when you're talking about somatic cell editing or in vivo editing. There you are likely going to be dependent on the use of engineered viral vectors. And there are a number of different viral vectors that are commonly used to deliver transgenic cargo that have gone through a number of different clinical trials and have pretty established safety profiles at this point. And the delivery vehicle that's sort of emerged as the most promising of all the different classes are known as adeno-associated viruses, or AAV vectors. The first AAV-based gene therapies were actually recently approved in the US for an inherited retinal disorder and also for a neurodegenerative disorder. AAV are small, non-pathogenic viruses that can infect a wide range of tissues, depending on what the AAV variant is. So there's actually a number of different ones that are all labeled very cleverly, AAV1, 2, 5, 8, 9. And depending on which variant you use, you can target with different efficiencies, different cells or tissues. And so we use these AAV vectors to deliver Cas9 and other genes into cells for gene therapy. And then the other question that's kind of implicit within that is, how do you ensure that the Cas9 is expressed or produced just within your specific diseased cell type within a tissue? And to do that, we rely on tools that are known as cell type specific promoters. So in order for a gene to be expressed, it needs to have a promoter to recruit the transcriptional machinery to produce the mRNA that then becomes the protein. And we can rely on promoters that work only efficiently within very specific or defined cell types. And those are known as cell type specific promoters, to restrict the production of the Cas9 protein to just within certain cells within a larger tissue. So we rely on engineered viral vectors for a number of in vivo based approaches. And then for ex vivo, we can use very efficient techniques based on or that are very efficient electroporation based techniques or other derivatives. So that's an excellent question, and it kind of depends on, like I said, the approach, the target, and the cells that you want to ultimately edit.
Okay, and you're still discovering more as time goes on and what this is capable of doing. So that is exciting. Dr. Hawkins, a question I have for you is more related to your experience in practicing medicine in other countries. And the question is, how does practicing medicine in lower income countries compare to practicing medicine in places like the US?
So yeah, primarily it is a much more clinical based practice. I will say this with a caveat that I haven't practiced in low and middle income countries for some time. I usually am mostly collaborating on research. But when I have, you're really working with in a more challenging environment in the sense that there are just fewer diagnostics. And that's not a bad thing, actually. Sometimes I think we probably use way too many diagnostics in the US and forget our clinical skills. So I can say that certainly clinicians I've worked with in low and middle income countries have much more skilled in the clinical skills department and are really sort of, think very clearly and focused on patients' care and use medicine wisely. So in addition to lack of diagnostics, there's obviously, usually medications are less, obviously available, usually choice. I mean, it depends what you're treating, but for instance, in hepatitis C, we have hepatitis C therapies readily available now to most groups of patients, whereas obviously in somewhere like Nigeria and Tanzania, where I work, there are almost no therapies available at the current time unless you have quite a bit of money to treat yourself privately. So.
About that. Oh, of course, if you have money, you can do the work. Thank you. Dr. Yunes, I'm going to ask you another question. And it was in regard to you mentioning that there was a black hole in the center of our galaxy. And is that anything we should be worried about?
Great question. So the center of our galaxy happens to be in the constellation of Sagittarius. And if you look at the center of the constellation, somewhere in the center of the constellation, there's a spot where all of these stars are orbiting around this spot. And we call that spot, the object that's at that spot, Sagittarius A star. Sagittarius A star has been identified as an object that has the mass of 4 million times the mass of our sun. So it's incredibly massive. Yet it's tiny, because if you point telescopes at that spot, you don't see the light of 4 million suns shining. However, you do see all of these other stars orbiting around each other. So the Nobel Prize in 2020 was given precisely to Reinhard Genzel and Andrea Ghez for doing these observations of the stars and the gas as it swirls and orbits around the black hole center of our galaxy. That prize was also shared by Roger Penrose, who was one of the people that predicted that black holes were a generic consequence of relativity. Having said that, the center of our galaxy is incredibly far away from us. It's what we call 8,000 parsecs. And a parsec is a ridiculously large astrophysical distance. So there's no way that the solar system will ever approach the center of our galaxy. We are very much on the outskirts of our galaxy. If you think of our galaxy as an ellipse, or it's really a spiral galaxy, it's an ellipse, and here's the center. We are like somewhere around here, close to closer to the edge. Whoops, yeah, the edge goes this way. Closer to the edge of the galaxy than to the center of the galaxy. So there's really, we are safe. I mean, if Earth or humankind will disappear, it'll be for other reasons, not because of black holes.
Woo, what a relief. Thank you. That is an interesting question, though. I think before tonight's presentation, I wasn't aware that we were anywhere within parsecs, let alone many of them, to black holes. Dr. Guy, one of the questions raised were in regards to CRISPR was, are there any concerns over CRISPR being used to alter human genomes in a negative manner?
Yes, there are concerns. And I think, so I mentioned one of the strengths of CRISPR-Cas9 was that it democratized the field of gene editing and really gave anyone with a pretty basic understanding of laboratory molecular biology science the ability to carry out gene editing. And so soon after the initial wave of papers demonstrating that it was remarkably efficient in cells, concerns were raised about rogue scientists or rogue actors using the technology to conduct what's known as germline editing, which is editing of germ cells to create kind of in a worst case scenario designer people. So potentially eliminating undesirable traits and engineering new desirable traits. And that's sort of the worst case scientific scenario of gene editing technologies. A couple of years ago, though, a scientist from China did publicly report that he used the technology to edit genes that are involved in HIV infection within the germline of embryos that then were used to produce viable children. There was pretty swift condemnation of the scientist and the approach, and a number of efforts have been raised to sort of prevent the use of technology for germline editing for the time being, until we learn more about really where that concept stands. I will say because gene editing is highly dependent on DNA repair pathways, it's becoming abundantly clear that we don't have a strong understanding of those repair pathways within human embryos and the gene editing outcomes that are introduced by CRISPR-Cas9. So while it is possible, and it's a serious concern, and people are developing regulations that we all hope to follow to prevent its use, we also have a pretty limited ability of carrying it out to the extent that we are able to use the technology in somatic cells potentially to treat disease, for example. So it is a serious concern. It potentially has happened. There have been consequences from that, and I think the scientific community is trying to come up with the best possible policies to police the use of the technology in the future, to study its performance within this embryonic context in human cells, and to ensure that it's only used under sort of the sparingly and if absolutely necessary in the future. It's not going to be happening anytime soon at any clinic or medical school.
Okay, thank you for that. So we're still in the learning phases, and we're going to rely on a system to be developed to kind of monitor it. Thank you. Another question, Dr. Hawkins. Can your Hep C work help in fighting the COVID virus?
So that's a great question. So there have been antivirals studied for COVID that have been used for hepatitis C cure, a couple that are in clinical trials, and there is a potential, put it that way, because there's a similar mechanism of action, but we have not seen any data yet.
Okay, and then as a follow-up question to you, Dr. Hawkins, I'm going to ask each of you, where do you see your field going in the future? Dr. Hawkins?
Well, I would love to see hepatitis C eliminated. I don't know if that's really possible. And COVID has put a little spanner in those works as well for now. But I'm also, my other sort of half is working on half of me is working on hepatitis B curative research as well. So that's another sort of big area of research, and I hope that in my career lifetime we do see some reasonable, a drug that could at least achieve a functional cure, and if not complete viral eradication. But we'll see. But there is hope for hepatitis, and certainly the monumental efforts that have been involved in getting a hepatitis C cure and the big steps that have been made there. There's no reason why that couldn't happen for hepatitis B as well. So I look forward to seeing that through.
Great, thank you. Dr. Guy, what do you see happening in your field?
From a basic science standpoint and a technology development standpoint, I think over the next 5 to 10 years, the CRISPR-based tools are going to be improved and become increasingly more sophisticated, safer, and more efficient. And from a therapeutics perspective, I think it's feasible that there is going to be the first gene or cell-based therapy involving CRISPR-Cas9 approved at some point. But I think just like for any other gene therapy, there's going to be probably hiccups along the way. I think this has already been demonstrated as a field that is a little bit volatile, and the public and media pay a lot of attention to. And I think oftentimes there are a lot of negative or positive reactions to either basic scientific data or preclinical data that can really influence the public perception of it. So I think overall, the field, I think, is really optimistic about the potential of the tool to enable new treatments and therapies, but it's going to take a long time. I think people are estimating that maybe within 15 to 20 years of when the technology was initially discovered, we can really begin to see the first transformative CRISPR therapies approved. I think we're still probably on pace for that. But I think people are very optimistic, cautiously optimistic, but with gene therapy, you never know.
Okay, thank you. Cautiously optimistic, good. And Dr. Yunes, where do you see things going in gravitational theory?
So the discovery in 2017 of the first gravitational waves are for sure opening up an entire new field. So right now, those two instruments, three instruments, one in Washington State, one in Louisiana, Baton Rouge, and one in Italy, they're offline right now, being upgraded so they can reach their design sensitivities. All of these observations were done with sort of an initial version of this advanced detector. In 2022, they should be running at 100%. 2022, 2023 should be running at 100% capacity. And when that happens, instead of getting maybe 50 detections per year, people are expecting maybe thousands of observations per year, allowing us to map all of the black hole mergers and neutron star mergers, not just in our galaxy, but in nearby galaxies, allowing all sorts of new discoveries. Like, for example, a recent one in 2017, actually, the same year as the Nobel was awarded, was the discovery of gravitational waves produced by the merger of two neutron stars. And from that observation, which was also seen electromagnetically with telescopes all over the world, scientists were able to figure out that most of the gold and platinum in the universe is really produced in the merger of these neutron stars and not inside the cores of stars like the sun, like Carl Sagan had told us. So we're not just the dust of stars. What was it? Was the saying was something like that, but we are also produced by the cataclysmic collision of objects that only Einstein and his contemporaries were able to envision.
Oh, goodness. Well, thank you very much. I'm going to stop asking the questions at this time, and I want to really say that we're appreciative of you sharing your insights on this exciting research. We are very delighted we were able to bring to the public information about why what the content was behind the prizes that were awarded in October, and helping us build our understanding of these fields, essentially. So overall, we want to say thank you for tuning in to today's program. Please go to c2st2 cnf.io to evaluate today's program. We do need your feedback to continue to offer relevant and exciting topics like this program this evening. If you'd like to rewatch or you've missed any portion of today's program, the recording will remain on YouTube, the YouTube channel, C2ST TV, and our C2ST Facebook channel. We extend our gratitude to Dr. Guy, Dr. Hawkins, and Dr. Yunes for joining us live this evening to share their expertise. And if you've enjoyed today's program, please consider donating to C2ST at c2st.org. We know on this day, Giving Tuesday, there's lots of demands for your dollars, but we would be very grateful in helping promote science and technology with the public. At our website, you can also find archives of other programs and timely STEM blogs. And overall, we just thank you again for watching. Stay safe.